metabolomics briefly, but not for long, don't worry, to focus on some metaproteomics methods. I've elected in this talk to be a little bit more surface about the actual science so that I can show you basically the kinds of questions that we're interested in pursuing with this methodology. And I have to start with some motivation, like why do we spend so much time and energy on the marine microbiome? So I'll start with that. And I've been testing out a few different motivations. We'll see which ones this audience likes. So my first motivation is philosophical. As many of you know, the surface area of the planet is mostly covered in water. Most of that water is salt water. Most of the life in that salt water is microbial. And so sometimes I like to say that studying marine microbes is the study of life on Earth. We can talk about that later and argue about it. But I also think that if you want to understand fundamentally how life interacts with the surrounding chemical and physical and geological environment, for this reason and for others, marine microbes are really not a bad place to start. And this photo was hanging up outside of my childhood bedroom. My parents are not scientists, but it's something that they did, I think, very well. And I'm grateful that that was true. On a less philosophical but more practical side, marine microbes do play a pretty critical role in the Earth system. So we talk about them sometimes for our climate system, for carbon cycling. This is one example of marine microbes playing multiple roles in what we call the biological carbon pump. So the simple story here in all these arrows is that marine phytoplankton are very numerically abundant. They do photosynthesis, which removes carbon dioxide, which is actually derived from the atmosphere. They convert that into microbial biomass, and then that biomass is kind of concentrated. It sinks more rapidly than a dissolved form. And this allows that carbon to ultimately fall into the deep ocean and be sequestered outside of the rapid cycling that happens between land, the atmosphere, and the surface ocean. So marine microbes are playing really critical roles in this carbon cycle and in many other biogeochemical cycles. And we know that their roles are also going to be changing as we grapple with climate change and also with long and short-term pollutant events. So we're trying to understand and constrain that variability now so that we can be better stewards of the Earth system moving into the future. So I like to show this video as an example of the kind of chaos that we're dealing with when trying to study the marine microbiome. This is a phytoplankton net tow that was taken off Catalina Island, so not too far from here. We drag a net for about 5 meters across a dock, so really not far. And there's tons of stuff in here. There's a starfish that floats by. I can't really see it, but this is the kind of complexity in this microbial ecosystem that we are now going to try to piece apart and understand the contributions of these individual microbes and the community as a whole to something like rates of photosynthesis, amount of carbon sequestered from that ocean ecosystem. And so if you're a numbers person and the video wasn't cool, I've been trying to constrain how diverse that marine microbial ecosystem really is. I think we need to update this number. I made this slide like 1.5 years ago. But basically suffice it to say it's orders of magnitude more diverse than many other biological systems that we study, like the human system or the human microbiome. This is in part not really a fair comparison because the marine microbiome is actually many habitats, many different types of habitats. The deep ocean and the surface ocean are extremely different from one another, for instance. But there's a lot of diversity there. And we're trying to capture some of that and also understand how that function works. So this is practically what our sampling and experimental approach looks like. We go to the sea and we sample marine microbes. We all gather around computers. It's not as cool as what they show in National Geographic. We look for traces of marine microbes and then we sample them. And there's many different ways to sample the marine microbiome. This is one of the more common ways. We take these big pumps, we deploy them to the ocean and pump lots of water onto filters to concentrate the microbial biomass. And then, so this is just an example of what that can look like. So these are filters from, I think this is from the Pacific Ocean, kind of in the tropical Pacific. And we see different sort of types of marine microbial communities, which you can see in the different colors here, actually, depending on depth, for instance. We also see differences in space and in time. And then we come home and we analyze those microbial metaproteomes by LC-MS. I'm not going to tell you a whole lot about the methodology, but we work on optimizing all steps of that process from sample handling, sampling with robotics platforms, for example, all the way through to the bioinformatics. And we face challenges all along the way. But yeah, so here's an example of some of the places where we sampled the ocean microbiome. This is just what I participated in directly. There's a broader community of scientists that have been doing this work across the world's ocean, including in some very big and important programs that are international collaborations. So what do we do with all this data? Because you get your mass spectra data, whether it's proteomics, metabolomics, you get Excel files or CSV files if you're lucky with lots of proteins or metabolite names and abundances. And then you have to figure out scientifically what to do. So that's what I'm going to focus on for the majority of this talk. What are the kinds of things we do with this data? So here's one thing you can do. We've been exploring using environmental proteins in sort of an inverse problem type of way. So we know that the microbial proteins are responding to some aspects of the environment. Can we use those proteins to infer something about what the environment is doing or what the chemical and physical properties of the environment are? So this is a protein from a marine cyanobacterium prochlorococcus, the little green dots. They're extremely important in the ocean ecosystem. And so oceanographers look at data weird. We plot depth in an inverse way. So the top of the graph is 0 meters surface, and then moving down, you have greater depths moving down to the ocean. And then you're also looking at data across latitudinal gradient. So from north to south on this transect that went sort of near Hawaii in the South Pacific Ocean. So we have the concentration of this protein, this prochlorococcus phosphate transport protein. We just had a really great introduction to nutrient transporters. And you can see that it's actually quite good mirror image of the concentration of phosphate in this region. So we have hypothesized in the past that phosphate is a limiting nutrient for the growth of this organism and is sort of setting the amount of photosynthesis it can do. And we can actually use its proteins in order to infer the chemistry of the surrounding seawater and also understand microbial strategies when that nutrient concentration becomes low. We can also do systems biology, and what's exciting about that for me is that we can resolve the systems biology in situ. So we often have a very hard time isolating environmental organisms from nature and bringing them into the lab. And if we're lucky enough to even get them in the lab, sometimes it's really hard to figure out how to design laboratory experiments to capture the complexity of nature, the microbial interactions that we know are playing an important role, the specific chemistry, the availability of metal chelators, et cetera. So it's really nice to be able to also, in addition to doing the lab work, study the organisms deeply into their biology in their natural environments where they've been evolving over time. So this is an example from two of my favorite organisms. They're marine cyanobacteria like prochlorococcus, but these guys also fix nitrogen. So they do this in a really interesting way. Crocosphaera, the little dotted one, does photosynthesis during the daytime and then fixes nitrogen at night. And it does this to avoid this problem that happens when you produce oxygen in photosynthesis, which will destroy your nitrogenase enzyme because it has very, like fragile iron sulfur clusters, basically. So Crocosphaera does a very normal type of chemistry or type of biology. It turns its proteome on in the day. All these lines represent groups of proteins. Turns its proteome on, wakes up, does lots of metabolism, goes to sleep at night, and turns on its nitrogen fixation proteins, which are the blue line showing you here. So you're looking at a 24-hour cycle. Its cousin trichodesmium, which is actually very closely related, also does photosynthesis and nitrogen fixation, happens to do both of these things during the day for reasons that no one can really understand or figure out how they even do it. But has this crazy pattern in its proteome where it's turning over groups of proteins on timescales of 2 to 4 hours or less. This is really crazy because this organism only divides once per day if you're lucky. So it's basically investing very heavily in degrading, synthesizing, and kind of juggling these two fundamentally incompatible metabolisms, whereas its cousin has decided, I'm just going to do this at night. It's really interesting. So then you could start to think and ask questions about why that has evolved in trichodesmium specifically. I'm not going to tell you why, but you can ask me later. It's kind of fun. We can also look at aspects of microbial ecology. So looking at how microbes interact with each other in nature. So this is that same nitrogen fixer trichodesmium. And these are all colonies of trichodesmium. So it's a colonial, it forms colonies in nature. And each of these colonies was sampled from the exact same phytoplankton net. So the same sample, so same environment, we would expect them to be similar, but you see there's huge morphological differences in these colonies. Part of that, it was known before. Trichodesmium makes these puffs, these like radial sunbeams, and also tufts, which are more organized, like in panel H here. But part of it was also interactions with metals. So we just had a really great interaction with our introduction to metals. Trichodesmium in some regions of the ocean actually interacts with other bacteria in order to acquire metals from particulate matter, which is basically dust that's raining down from the atmosphere. Those particles, you could think of them like grains of sand, grains of salts. They contain a lot of metal and also phosphorus. And trichodesmium uses that in order to fuel its metabolism. And what we can see is actually that morphology, those interactions with microbes, interactions with other, with these dust particles, is imprinted on the proteomes of individual colonies of trichodesmium. So this is an iron-containing protein ferredoxin, which is more abundant when these particles were interacting with dust and other organisms in nature. But you can see there's also a huge amount of variability. This is like a bad way of looking at the data, is what this bar plot is, or box plot is showing me, which means that there's a lot to unpack also about heterogeneity in microbial ecosystem structure in the ocean. And that's going to be a really important place to start thinking about methodological challenges moving forward. We can also do a little bit of in situ biochemistry. I call this more of our hypothesis generating side of the work. But this is another example of a marine photosynthetic organism, Synechococcus, also very important. And this organism, along with others, makes this enzyme alkaline phosphatase, which helps it to acquire phosphorus from organic matter when dissolved forms of phosphorus are very low in the environment. So this is kind of a secondary way of acquiring a rare resource. And this organism has many different alkaline phosphatase enzymes. This is one example, Pho-A, which is thought to be a zinc-containing protein based on homology to E. coli or Bacillus, because that's the best we can do often in terms of identifying the proteins of these marine microbes. And what we found actually was that when we took natural populations of Synechococcus, brought them onto the ship in mesocosm experiments, so big pots, and added nutrients to them, what we saw was that indeed, when we add zinc, we do get more of this protein, suggesting that it is a zinc-containing protein. But also when we add cobalt, which is a very similar metal, we get a very similar, actually higher amount of this protein. And we actually think that based on this work and work that we're following up with, that this protein might actually be a cobalt-containing enzyme in nature. And this is because zinc is very, very low in the marine environment and cobalt is quantitatively more abundant. So maybe they figured out how to substitute these metals. Okay, so we can do all these cool things. I think there's a lot of things that I would like to continue to do. And I wanted to highlight some challenges in metaproteomics, I think specifically ocean metaproteomics, but this will be true in any metaproteomic experiment or type of system. The first is that we have very complex spectra. So it's really fun to go to ASMS and talk to people about their very complex HeLa sample. And I'm like, yeah, that's very cute. Look at our spectrum. So we have like very, very large numbers of peaks in our samples. This is DDA data. This isn't even DIA. So as we move forward into the DIA world, it's going to get even crazier. All of the peaks that are present in those spectra are often lower in intensity, partially because we have more diversity. So we struggle to acquire MS2 spectra of all of the peaks, for instance, in DDA mode. And we have a dilute sampling issue. So what we're often doing is taking hundreds or thousands of liters of seawater and condensing them onto a single filter and doing the metaproteome of that. Microbes don't live and experience 200 to 1,000 liters of seawater. They experience, maybe 3 maximum cell widths around them. So we're trying to understand how they work, but we have to integrate a lot of that heterogeneity into these samples. So I think we have a lot of way to go also in our sampling efforts and in thinking about how to get more from less biomass. And that's starting with sampling through to sample handling and even our mass spec work. And then partially the reason to pursue that particular type of work as well is that the ocean is very patchy. So this is a satellite image of the South Atlantic Ocean, and you're looking at kind of kilometer scale variability in cyanobacterial blooms that we're going to drive boats across and try to capture aspects and build up into global ecosystem models. We need to be able to understand that patchiness. And we need to do this at scale. So it doesn't, we can do a lot with local sampling, we can do a lot with single expeditions, but if we want to go to a place where we can actually make recommendations or constrain variability in the ocean ecosystem, we need to be doing this as a community of scientists, and we need to speed up our analyses. So this is something that we're working on as a community, but we quickly approach clinical cohort size sampling efforts. And of course, we don't necessarily have the resources of our clinical counterparts. So we're trying to figure out how to do this in the scrappy way is sometimes how we say this. And so for this reason, we're really excited about developments in mass spectrometry technology and bioinformatics and robotics. I'm highlighting one platform here, but I think all the developments that are happening now are really exciting. We're able to now get 20,000 up to 100,000 proteins identified in our sample across microbial community structure. And these numbers are boring to me. I'm interested in the scientific questions, and those questions are being addressed. Now we have the capability to address, to do ocean metaproteomes on 30-minute gradients now from less than 50 nanograms of material, nanograms of material, whereas before we were working with 10 micrograms of material and 8-hour gradients over 2 dimensions. So I think that this is without trying very hard, frankly. This is just taking what's already been out there. So as we continue to try and apply methods, I think we're going to hopefully be able to address some of these questions. And for me, this is really important. I've been thinking a lot about these metaproteomics data as records of environments that are changing rapidly in time. So we're trying to collect this data and digitize it and make it available to more researchers so that we can mine and do retrospective studies into the future. I wish I could go back 20 years in time and look at what Trichodesmium's metaproteome was doing, or even its metagenome. Can't do that, but we can start providing for the next generations to be able to do that. It's also cheaper than having a bunch of -80s. And I'll leave it with one last thing. This is from my friend Jackie Saunders, who's at UGA. So this is each flash here is an individual protein that has been identified in a transect, actually in the South Pacific as well. And you're looking at depth again on the y-axis and transect on the x. And then it's a heat map basically of the protein abundance. So each one of these little slides is the beginning of a scientific question or story. No one lab can ever answer all the questions that are coming out of this data. So we've been trying to make it available. We have some portals to look at that. But I think if anyone's interested in looking at this data, we're very, very, very happy to have more help looking at the ocean microbiome in this way. Great. So I'll end there. I just started at USC in January, so not too far away. And we'll be doing this kind of work. And I have 2 great graduate students and looking for more people if anyone's interested in doing some ocean metaproteomics.
So working on the microbiome planet, then after the microbiome you mentioned that you mentioned in the microbiome cells. So what do people try to understand across all the microbiome for the
Yeah, so I mean, we focus on we tend to focus on the internal proteins that are in the host. So we're not always thinking of a kind of public good type protein. But we do have species and subspecies and strain level specificity that we can do depending on which peptides we choose to look at. So the way we do this is we can kind of coarse grain our peptides. Some of these peptides will be shared because of homology to other organisms. And then we can kind of decide at what level we want to probe that question. We can say, I want to know exactly what that strain makes. Then you focus on those peptides. And you can do that. Or you could say, I want to know all of the alkaline phosphatases from all of the phytoplankton, that you could focus on peptides that are more shared. Does that kind of answer the question? Yeah. But yeah.
How many proteins do you think you're missing? Because there just aren't homologous sequences that you can mimic.
So for instance, we had, so that 88,000 proteins, those are only the ones that had some KO annotation. So first of all, we're missing a lot of peptides because the peptide spectrum matching is low. Partially because some of those spectra aren't peptidic and also partially just because algorithms struggle with that. Then we have to match our peptides to proteins. That's also kind of tricky when you have multiple organisms. Like, how do you deal with the ambiguity there? And then you match proteins to like function, and it's often like less than 10 to 20% will have that kind of annotation. So we're in full struggle mode there, but I try to be positive about it at the same time, because at the same time, like we could put a lot of energy into figuring out every single thing, but we also are able to actually address a lot of science questions with what we have. So, but yeah, good question. Who even wants to work on that?
I have a follow-up question about the sequencing depth. And you just answered my question, like how to match the protein sequences to different species. But since there are so many different species in just 1 ml of seawater, and how deep you're going to sequence those peptides in order to cover as much as possible of those proteins from different species?
Yeah, so it depends on which kind of ocean microbiome you're in. So sometimes I think of the ocean microbiome as having a dominant proteome. So it's not like blood plasma or something. We have a dominant protein, but we often have a dominant organism present. But that protein will have its 5,000, protein encoding genes or something, and we can capture more than 50% of those typically, which is probably a pretty good number. And then we have lots of other complexity, like 20% of the microbiome will be lots of other things, and it's all the other things. I think it depends on the scientific question. I think if you want to do you want to scale up to quantitative carbon cycling, maybe you need to just focus on the dominant organisms. If you really want to dig deep into like fine scale microbial ecology, then you need that deeper taxonomic coverage of the proteome.
Do you need the help of the metagenome data in order to have reference genomes or whatever if we don't know anything beforehand?
Yeah, so with the proteomics, we match our spectra to a sequence database of possible peptides in our database. And we get that typically from metagenomes like transcription, which we often work with collaborators to match metagenomes.
Just one more is about the annotation, but I think that's an issue. Like how do you guarantee that your protein annotation will be accurate, or there's some other issues about that?
We're relying on the metagenomic annotation. Yeah. So we have to trust our protein.
Thank you.
Good question. Yeah.
A fascinating topic. Do you have any insights in the abundance of like the proteins inside of cells versus the abundance of proteins that are secreted in the ocean?
So yeah, the proteins that are secreted are a lot lower, like orders of magnitude less. We have to concentrate a lot more.
But in the total ocean,
Like in
where's
terms
most
of
of the
what?
biomass?
Like where is the biomass? It's probably marine snow. So there's like flocculants of organic material. So these phytoplankton kind of die, but they get eaten and fish poop, and then it makes this marine snow, which is like, you should look up videos of it. It's super cool. It's probably like the proteinaceous material is probably in those kinds of materials, derived from the phytoplankton growing in the surface oceans.
Do you have a good handle on analyzing the secreted proteome as well?
So I haven't personally worked on it very much. There are people that have. It's a concentration issue, but I think that a lot of the proteins. So anyway, there's ecological things going on because microbes will often tether a lot of their excreted proteins, because if they release that, they'll just dissolve away. And ocean microbes are quite dilute. Know that we are often missing those proteins that are stuck to the membrane because we use them right of extract.
All right, well, let's thank Noelle and all of our speakers again. Take a 20-minute break. Then we'll come back to the next session.